Hello to everybody for joining this second session of the Virtual Ampha Academy 2021. I am very glad to present you the new Ampha P20, the next generation and pollen analysis and the newest member of our Ampha family. So before I do this, I would like to briefly introduce you into the technology, what is behind it, spend a few words on the Ampha Z32, and then, of course, I will focus on the characteristics and features of the Ampha P20, as well as presenting you some accessories which come with the instrument. Finally, I want to spend a few words on the positioning of both instruments. So the Ampha P20 in comparison to the Ampha Z32, where both instruments are used, what are the areas of application. So most of you might know this instrument here, the Ampha Z32, the pollen analyzer, the current instrument. It is based on a technology which is called impedance flow cytometry. And this technology enables to characterize the electrical properties of cells. So not only of pollen cells, but also animal cells, human cells, bacteria, yeast, so many other more applications. The big benefit is that this is a label-free technology. So you don't dye these cells. They are not altered by the measurement. So they remain unchanged before and after the measurement. The whole measurement takes place that the cells are suspended in a buffer, in a specific buffer, which is developed by Amphasys. And it is measured in this microfluidic chip, what you see on the right-hand side. And as a result of the measurement, you will see this scatter plot. Every dot in the scatter plot represents one cell. So what you have to do is then you have to make this gating, so determine which are the dead cells, which are the viable cells. You see the viable cells on the right-hand side, the dead cells on the left-hand side. And you can see more or less in the minimum where the line between the two populations lie. So now you will ask, how do you know that the different kind of cells, I have added to the viable and the dead cells also the aberrant cells, so to have a third kind of cell, how do they get to the specific position in the scatter plot? And how do you know where to put the gatings? So all is based on the microfluidic chip. The microfluidic chip has a channel, and the width of the channel is somewhat bigger than the average of the diameter of the cells. So the cells pass through this channel, and the channel has two electrodes which generate an electric field. So now that the cells pass through the electric field, depending on their nature, they exhibit a signal which is then detected and which depends very much on the properties of the cell. And these parameters are, first of all, the cell size, which shows you a different behavior, and also the properties of the membrane expressed as membrane capacitance. This is also the second parameter. And third, it is the internal of the cell, so the cytoplasm conductivity, which at the end determines where exactly in this scatter plot the individual cell will be placed. So you know this instrument, the Z32. This is our high-end lab instrument. This has a very universal application, and you have learned already in the previous session that we can measure more than 200 different crops. And normally you determine the pollen viability and the number of the pollen cells, but this instrument also allows you to determine the microspore developmental stages and also the pollen ploidy. So what are the benefits of using this technology in the seed and breeding process? First of all, it's all about efficiency. You will save time, money, manpower, and you will get a lot of information about your plants. So the application spans from research over the whole development cycle up to seed production. In research, for example, you can optimize your processes when you are looking for the selection of microspores for the DH protocol, for the DH production. It is possible with our technology to determine the right stage of the microspores in order to proceed for the DH protocol. And as mentioned already before, my colleague Federica, she will give a talk tomorrow exactly about this topic. Then in breeding, as Dario in this previous session mentioned, it's about precision phenotyping. So it is to know all the information about your lines. It is to systematically screen the lines to find out which lines are heat tolerant, which lines are the best lines, best male lines to give the most pollen, and other information like that. In production research, it is very important to optimize the cost. So if you can reduce the male to female ratio, then you can reduce your cost in growing the plants for the pollen production. You can also improve all the process if you know when is the best point in time to collect the pollen and not to spend manpower on collecting pollen, which is not sufficiently viable. In seed production, finally, it is important to determine the quality of the pollen before and after storage, before and after transportation. And our technology allows not only to determine it in a very high accuracy very quickly, but also completely location independent. So with 2 independent instruments in 2 different places over the world, you will obtain the same results. So that was enough about the general technology and the Z32. Now I want to focus on the Ampha P20, our new instrument. We are very proud of that. And the whole development that started with the feedback of our customers. And the customers having the Z32 in use, they had 3 main issues where we could improve. So one was the portability. The second was the ease of use. And the third, to simplify the data analysis. As you have seen, you have to place the gates by yourself, and you have to know how to do it. So as you can see from these pictures already, the portability is something what we have achieved. You can use the instrument, of course, in the lab, but it is made for outdoor use. So if you want to use it in the greenhouse, in the field, this is the place where the instrument is designed for. So I will give you a few of the most important features of this instrument. You see a sketch of the instrument, and one of the most important features is the crop-specific chip. I will talk in detail about that later on. So the touch screen is also something which makes the whole handling of the instrument more easy. With a touch screen, it is possible within a few clicks to start the instrument and get the measurement done. The touch screen also has a very intuitive workflow, and behind the screen there is a processor, which means you have the whole computer inside. You do not need a second computer or an external computer to run the instrument, to store the data. This is all inside the instrument. As a result of the crop-specific chip, if you insert this chip, it will automatically load all the settings which are necessary to make the measurements. And another result of the crop-specific chip is that the data analysis now is completely automated. I will also talk in detail about that later on. It is possible to download the data. We have a USB port. You can download the data via Wi-Fi. Once the data are collected with the instrument in the instrument, you can download it to your local internal network. The dimensions are such that it fits as a hand luggage in the airplane. You can carry it on in the airplane. Also, the battery allows that. It has about 8 kilograms, and you have seen it's quite a protective case. So it's a robust external shell. And finally, the batteries, they are exchangeable and also rechargeable. And that gives you autonomy of 3 to 4 hours. And we offer spare batteries, so you can work the whole day outside the lab, in the field, and make your measurements. So portability, I've mentioned, this was one of the most important features, and you have seen you can do the measurements in the field, in the greenhouse. You see this picture where we have taken it to a wheat field. And this is one of the most important, well, one additionally very important aspect. For the first time, it is possible to measure the viability of pollen of species which have a very short lifetime. So for example, wheat and corn, they have a short lifetime. We have developed the chips for these species, and rice would be also on the short list for the next applications. So instead of bringing the pollen to the lab, you can bring the lab to the field and do the measurements on the spot. So in the case of wheat and corn, the viability is not changed between taking the pollen up to the measurement. You can do it more or less instantaneously. Then the ease of use. So with the touchscreen, we have a very easy menu here. You see on the right-hand side, the main menu, which is very easy to follow, to operate. It is user-friendly, and with this, it also allows to do a lot of routine measurements and systematically screen a lot of lines, a lot of measurements in a very short time. Some examples for these, for the menu, for the ease of use is, for example, this is the main menu. So if you start the chip control menu, it will tell you that a tomato chip is inserted. You will measure the viability, and it also allows you to see how many measurements are done. If you then go to the measurement screen, to the measurement menu, that will allow you to run the measurement. You will see the progress of the measurement within time. This is quite fast. it's more or less 1 minute, the whole measurement. And immediately after the measurement has stopped, you will see the result on the screen. So this is really a big improvement, that you do not have to do the gating on your own. It immediately shows you the result on the screen. We also have a maintenance menu. This is not necessary in order to do the rinsing between two measurements, because this is included automatically in the program. So after each measurement, it will automatically rinse. But if you want to do a disinfection or an additional cleaning, then this will be possible with this menu. And noteworthy is this. If you are used to the Z32, we have an automatic chip deblocking program. So once the chip should be blocked, it is possible with this menu, with this program, to deblock the chip. And as I said, for the data transfer, you have also a menu and a selection of possibilities in order to download your data. What we also have installed are wizards. So that will explain you how the instrument is set up, how you insert the chip, how you do the cleaning, all the information which supports your activities working with the instrument. This you can find all in the wizards in the menu of the instrument. So as I said, the automated data analysis is, for me, one of the most important features of this new instrument. This is possible due to the fact that we have developed crop-specific chips. And that means that for each of the species mentioned below, tomato, pepper, corn, and wheat, for these crops, we have developed a chip already. So we have measured hundreds and thousands of measurements of samples in order to develop an algorithm, and based on this algorithm, then develop this automated data analysis. And this allows you to very quickly see the result of your measurement. So this is how the whole, how the screen looks like. You do not have to do a manual gating anymore. So it is very fast. You have immediately the results on the screen. If you know that your threshold for pollen viability is 60%, here you see 81%, and you can continue with, for example, the collection of the pollen. It is very easy. You don't need to know the interpretation of the plot, where to put the gates. So it simplifies extremely the handling of the whole instrument. And what is also very important, even unexperienced users can work with the instrument. It is so easy to use based on this automated data analysis. As I said before, you can download the data. So you can see the scatter plot on the screen, but if you want to really post-process the data, which is possible, then you have to download it via USB or Wi-Fi. And we provide the software to do that. So then you need an external computer, your laptop, with the software, and there you can post-process all the data. We offer an accessory case, and this has the same size and the same design like the instrument case. It contains all the material and buffers and consumables which are necessary to run at least 100 measurements. They are all can be replaced, refilled, and so you can take all the material which you need also to the field and do the measurements there. We offer a battery charger. We offer spare batteries, and also a barcode scanner so that you can read immediately your sample number and include it in the instrument. So finally, about the positioning. The Z32 offers the full flexibility, is the high-end lab instrument, and covers all the pollen of different crops. And in addition, you can also measure microspores and ploidy. So with that, it is the ideal instrument for research, breeding, and production research. The new Ampha P20, this is perhaps less for the research applications because it doesn't have this full flexibility. We offer tomato, pepper, wheat, and corn chips at the beginning, but it has the advantage to have a full portability. You can very easily, very fast measure all your samples, do a systematic screening, and due to the automated data analysis, it can also be used in production and can be operated very easily. As a summary, we offer one technology, two instruments, and many areas of use. And if you use the pollen analyzer, our instruments, then you can improve your process efficiency, you can speed up your breeding process, and you can save resources, may it be money, manpower, or pollen. So thank you very much for your attention, and I'm free to answer all your questions. Thank you.
Thank you very much, Jörg, for this clear presentation and for this nice overview about the P20. I went through the many questions that came in, and it seems that there is a lot of interest about it, and still there are coming in new ones. I tried to kind of sort them out in terms of looking for the more commercial ones and the more technical ones, and start with the commercial ones. One is, for example, will there be a support and maintenance contract for the P20 as well?
Yes, there will be a support and maintenance contract for this instrument as well. Many of the Z32 users, they know it already. We are finalizing these contracts and will offer them soon.
Okay, thank you. Then one attendee wanted to know whether it is already planned to develop new chips for other crops, and if there is already something in the pipeline.
Yes, we have a short list already, and we have received a lot of interest for new applications already. So there are in the vegetables, brassicas, cucurbits definitely, but please feel free to send us your wishes. We are happy to receive your comments, and definitely we will get in contact to you in order to know more about your specific needs. And this is the same for the row crops. I mentioned rice already. That would be an ideal candidate because it also has a very short-lived pollen. So please feel free to send us your wishes and your comments.
Okay, thank you. There's another question regarding portability and traveling. We were asked whether we foresee some airline travel issues with the P20.
No, we don't foresee, because we have chosen the battery exactly in order to comply with the flight regulations, and from the size it fits to in the hand luggage. So we have been traveling with the instrument in the airplane. We haven't faced any problems.
Okay, thank you. Now I go rather to the technical question. There were a lot of technical questions actually, and let me see. Yes, if I can answer them. For example, we are asked whether the P20 gatings are preset in a default position and how this was determined. And you already mentioned it a little bit. The algorithm or the gatings that are preset were calculated according to many hundreds or even thousands of measurements. So they are fixed. They are not being, they don't need to be changed, and they work properly for almost any lines of the specific pollen species. For example, the tomato pollen chip will work for actually all the lines we have tested so far. And this is because we also developed a new buffer for this instrument. And these buffers give a much higher robustness of the measurement. That's why it works quite well. So this is also an answer to the question whether we have different buffers for the P20 and C32, which is in fact the case. The P20 will have new buffers, and you can not only use the P20 with those new buffers, so you cannot use the old ones. Another question was whether we have tested ornamentals already with the P20. But you can answer it.
Yes, we haven't, let's say, we haven't developed a chip, and therefore it is not for use for ornamentals so far. We would need to develop a specific chip for ornamentals in order to do it.
Exactly, but this is just a matter of testing and optimizing conditions. We have another question regarding, let me see. Yes, if you mentioned already, one people was asking whether we can use a barcode scanner. Yes, it's possible, as you mentioned. And we are also asked whether it is possible to display the scatter plots on the touchscreen and whether it's possible to edit them. And here I can answer, yes, you can display them on the touchscreen, but you cannot edit them. You can just have a look whether I mean, this is mainly for the C32 users, which are used to look at the scatter plot, and you can compare it, but you cannot basically edit the scatter plot. You can, however, transfer the data from your P20 to an external laptop where you have normally running your Amphasys software, and there you can edit the data and look at the data as well. Another question was asked whether it is possible to connect the P20 with an autosampler. And here I can say that it's not planned to use the autosampler with the P20. It's planned for, let's say, the normal lab automation, applications and for walkaway solution, but not for the field. And another question was asked regarding the C32 tubing, which made some, we had some issues with, mainly with the silicon tubing. We have removed the silicon tubing, as you have possibly seen from the picture, so we don't have to replace them regularly, and we don't see the need to open the instrument, as was asked, to replace the inner tubing, because this can be done in the normal maintenance and service activity. I had another question regarding, now coming, and a question regarding, yes, it goes in the same direction. Is it possible to change the gate on a P20? No, it's not, but you can transfer the data on your laptop and change it there. But as mentioned, only for the 4 crops that are currently available. Another question is, yes, it's rather directed to you, Jörg. How much does it cost, I guess, the instrument?
Please send us a quote for request. Due to the fact that we have different currencies, that import taxes in countries vary and differ, therefore it is not possible to give one number, and therefore please ask for a request for a quote and we will answer that.
Okay, another question is, regarding ornamental crops, whether it works for petunia, marigold, vinca, I can just say that we tested many ornamental crops with the C32, not with the P20. And normally we find conditions that work properly for the C32. The question is whether it makes sense to automate it for the P20. And this is also something that we can discuss if you just call our support, for example. Another one or two questions. I was asked whether the instruments need to be calibrated and how often. And this is not the case. The instrument has a self-calibration mode in which we calibrate basically before every single measurement. So there is no need to calibrate it. It does it because it's programmed already for that.
Sorry, Marco, we have time for one, maximum 2 more questions. Thank you.
Okay, let me see. This is an interesting question. Is pollen viability always highly correlated with pollen germination capacity? Well, we have done many measurements with the 4 crops that we have shown, but also with other ones. And normally one can say if you have a high viability, you have also a similar germination capacity. The question is rather whether you have the best germination conditions. We have found basically that you have to figure out the right buffer for your germination, otherwise you will get lower values. But there was an Ampha Academy talk last year in which we have focused on that topic, and we can of course give further information if you, for example, contact our support. And let's see whether I have a final question. This is, what is the buffer for analysis? I'm sorry to tell you that we cannot, of course, disclose it. But as mentioned, the buffers for the P20 and for the C32 are different. If you can, however, use the P20 buffer for the C32. For those users using the C32, they must be aware that they have to change probably their gates and their templates if they use the newer buffers for the P20.
Okay, so thank you very much, Jörg, for the presentation. And also thank you, Marco, for answering the questions. I have seen that some questions remain unanswered, so don't worry because we will be sending you an email. Also, if you want to receive more information, if you want to receive a quote, if you want to receive an online demo, please write to us at info@amphasys.com. Also, I remind you that the session has been recorded. So very soon you will receive a link, and from this link, you can download the video. You can either watch it again or share it with the colleagues that were busy and that couldn't attend today to this webinar. And also, when you will close this session, you will receive a questionnaire. Please take a couple of minutes to answer because this will help us to improve for the future. And also, you are interested to answer it if you want to inform us about the new applications of the P20, about the new crops. And now we are closing this second of the 6 sessions of the Ampha Academy 2021. But I want to invite you for the third one, which will be the last of today. We have received in this session and in the previous one a lot of questions about microspores. So if you are interested in knowing how the French Institute Begenov is using the Ampha Z32 to study with microspore for DH protocols, please stay with us. We will be starting in around 20 minutes, the third session. So thank you very much and see you there.